But anyway, so I will talk about this, the Encyclopaedia of Domains, and also hopefully get on to at the end some recent work that we've done as kind of an application of this, of a downstream of how we can search for structurally similar multi-domain proteins using that resource and some simple algorithms. So of course, you know, this is one of the first times, I guess it's post Nobel Prize thing to say, I guess. Nobel Prize in chemistry, because everyone knows this. It doesn't make any difference to what we've done. It's just interesting that this is the kind of literal background to the data set that we're using. For protein structure prediction, of course, Demis and John take the share of that, and the other half for protein design with David Baker, of course. I think that's something we always long predicted. I think it's kind of interesting. I was involved with AlphaFold in the early days. I mean, I met them in 2016. I sort of they hired me as a consultant. I was the only bioinformatics, computational biology person on the team at that point, and I wasn't involved with the later stuff. Sadly, I didn't get a medal to in Stockholm, but that's the way that science works. But it's quite surprising that literally lit the blue touch paper in 2016, and then a Nobel Prize gets awarded in 2024. I mean, that is quite surprising. I did joke, because obviously after the thing was announced, my line manager at DeepMind at the time emailed me and just said, "Oh, isn't this fantastic news?" And I joked. I said, "Well, I should have really put a clause into my consultancy contract saying that if they got a Nobel Prize within 8 years, they'd give me a $1 million bonus." I kind of think they might have taken a risk on that, given the probabilities, or they might have considered that $1 million was actually quite a reasonable amount of money to pay for a Nobel Prize. I don't know, but both ways. But I didn't do that, and congratulations to them and the team. And I'll talk a little bit about AlphaFold as a background. Obviously, an outcrop of that, of course, is the data set that we've used, the AlphaFold Protein Structure Database at the EBI. And I guess there are 240 million protein structures covering most of the UniProt entries, minus things like viruses and some much larger chains and a few things like that. Those gaps may or may not be filled in the future. We await updates of the database. But for now, this is the data set as it stands. Now, most of the structures, this is kind of the trigger for this, was I guess many of these structures look very much like these things I'm showing you here, where you can see very well ordered protein domains embedded in what we call generally orange spaghetti, I guess, or loosely the term for this kind of thing. We know that protein domains are the structural and functional evolutionary units of protein, so identifying them has a high value. Trying to do this by hand is tricky. I mean, I've tried to do it, and it's a lot of effort just to do one. And so that's where we bring into this project the Encyclopaedia of Domains, and I'll tell you a little bit how that project started off and how that's developed. Now, AlphaFold itself, I guess, has made a big difference. I always like to wind it back a little bit, because sometimes there's a little bit of overhype about what AlphaFold has created. This is showing out, you've probably seen a plot like this before. This is a plot showing the accuracy of models in the CASP experiment that's been going on since the beginning of my scientific career. I've been involved in it in one way or another. Not this year, funny enough. The last time I wasn't really involved. But this has been every 2 years. We've evaluated the state of the art of the field. And what's the big selling point here? I'm not going to talk about this in detail. Was that this is the CASP14 results, and it's just showing the huge impact that adding the AlphaFold results had to the average result from this CASP. But the interesting, what I think I'll point out is that this is for the hardest category, and there's a huge impact there, where we're producing very accurate models with AlphaFold and less accurate models without it. But if you go to the easier category, and what we mean by easier are things where there's detectable homology to something in PDB, and you can use comparative modeling tools like Modeler, Swiss Model, and so forth. Actually, AlphaFold hasn't added a lot. And that's not a criticism, I'm just saying that is the situation. We were already quite good at modeling those proteins. AlphaFold has arguably made it a little bit better in some cases. It has also made a larger, but not as large improvement in the area of fold recognition, recognizing very distant homologs or analogous structures. So this is just to kind of temper things. And I guess this slide here just shows, again, this is me on my soapbox. All the press releases that came out were saying things like, well, AlphaFold has created now 1,000 times more structures than we had before. And that's, of course, comparing PDB with what's in the AlphaFold database, which essentially was large subset of UniProt. But in reality, if you actually run something like the Gene3D tool from or resource that the CATH team have developed, this shows that actually we actually knew quite a lot of structural information in UniProt. So the blue bars here just show you how many chains in the AlphaFold database actually have domains that could be detected by the hidden Markov model sequence-based methods that Gene3D uses, and this is compared to the whole of the AFDB. And if you look at that, then, and this is actually just looking at the TED version of this now, this is showing, so this is not changed, this hasn't changed, it's just showing that actually in TED, the majority, even all of the chains in the AlphaFold database have at least one detectable domain using TED. So this shows the jump between sequence and structure, but it also gives a more realistic view that the structural picture has only gone up by, say, 50%, and not the 10,000% or whatever or whatever number of times it looks like in the naive view here. So it's just to say that it can obviously be overestimated how much of a difference AlphaFold has made. But obviously we've got these structures in a database, and that's probably the big difference. So this is the paper that came out. So I mean, obviously, we were pleased to get into science. It's a nice paper. I mean, Christine and I have been working on collaborating together on and off from the beginning of my career, certainly, where we were both in Janet Thornton's lab, and we were doing things in those early days like threading. And I remember threading through all 102 different folds that we had in PDB at that time, and being quite impressed that I'd managed to find all these things. And that led on to things like trying to count the number of folds. PDB was expanding exponentially at that point, so large numbers of structures coming in. And we started talking about things like, trying to count the number of folds. We failed to do that, but in failing to do that, we defined things like the superfolds that are defined in CATH. And eventually, over slices of cold pizza at a railway station, Pizza Hut, I think it was, we hatched a plan to develop the CATH database, and maybe we could systematize this and actually catalog all the folds and superfamilies and so forth. And that's what CATH stands for: class, architecture, topology, and homologous superfamily, which is a name that we hatched that day. So in some sense, it's inevitable that a paper like this would emerge eventually. We sent Janet Thornton an early copy of the paper. She said, "Oh, this is marvelous. This is what I always had in mind." And so this always looks like a carefully hatched plan. In fact, it wasn't quite so carefully a hatched plan, as I'll tell you as I tell you what really happened. So this is the team. I'm going to show you the pictures now because I'm sharing slides other people have prepared, but we kind of had to make sure that everyone got a mention. We have the little pictures on many of those slides, and so this I'm sure you're going to memorize all these names. I'll be asking questions at the end, but don't worry, no, I won't. And I'll have the names again at the end. But this is just so when faces arrive, you can say, "Oh yes, that's so and so." But if you get a copy of these slides and go back and read them, you'll be able to do that. I've tried to make this, so that it's not. So I've done the sort of thing they do in Hollywood. So Christine's on the right, but higher up. I'm on the left, lower down, to try and make this kind of that it was very much a 50-50 thing. We all contributed different things to this, and I'll tell you how it actually started. This all looks like a very well hatched plan, but it kind of didn't work out quite that way. The TED highlights, and I'll get to a bit more of this as we go along, but just so you know, essentially we took the AlphaFold database. We do lots of AI-based, it doesn't have to be AI-based, but it turned out that's what a lot of people were using, AI and machine learning-based domain segmentation, that's automated rapid approaches to dividing the domains out of these structures. And that formed the master TED file now, which has 365 million domains in it. And then the simple job of labeling these things with matches to CATH superfamilies and folds, not so trivial, but as we'll see. And then maybe looking at the more interesting stuff at the end that wasn't immediately labelable. And we'll look at a few of those examples. So this is a kind of spoiler for what you're going to hear. So again, I've told you about the AlphaFold database. This was the thing that kind of kicked off this work. It arrived in various tranches, literally just a few model organisms, and then SwissProt, and then eventually the whole of UniProt. And we had an interest in my lab in analyzing this data on a large scale. We were particularly interested in domain interactions on multi-domain proteins and improving the modeling of multi-domain proteins. And of course, the problem was that when you look at these things and download the whole 23 terabyte chunk of data, this is what a typical multi-domain protein looks like. There's interesting stuff here, packing of these domains, and this is the thing that we were interested in. We were trying to improve this for the modeling, or trying to develop a resource that would help us improve the docking, if you like, of these domains. But of course, we had to kind of throw away all of the potentially disordered regions, or just regions that AlphaFold wasn't able to fold. And so we set about developing a tool for doing that. Andy in my lab at the top there said this is an interesting problem. I'd be happy to do that. I think he was still happy at the end, but we'll you can ask him. But anyway, so we set about doing that. We decided to use technology that was kind of like a reverse version of what was in AlphaFold. So we used invariant point attention, which is an SE3 equivariant model, which means it takes into account not just distances but rotations in space. And that was a very powerful part of the AlphaFold, of AlphaFold 2 at least. But you know, it was, we thought, a reasonable start. I'm not going to go into the details of this. I'm just going to say you can read the paper at the bottom there. There's a few highlights of things that we did slightly differently than other people, but essentially it took a lot of effort to get it to work as well as it did. It actually ended up being very fast and very effective, and eventually got into a paper in this Nature Communications paper. Sean here was kind of the person who kind of had the need for the domains, and also was involved in this paper. Now, what happened, this is how the TED project started, and before the paper, I think, was finished, I can't remember exactly when, it turned out that Andy came to me and said, did you know that someone in Christina Ringo's lab has been doing exactly the same thing as me? It wasn't quite exactly the same. It was, I mean, eventually it turned out. And of course, you know, that's fine. I mean, you know, we work in the same space. We share lab space in the same area. But, you know, obviously it triggered a sort of, you know, thing, well, okay, it's a bit of a waste of resources if we, but, you know, if they knew that we had this thing already, maybe they wouldn't have bothered, and we'd know they had it, maybe we wouldn't have bothered. And then there was a little bit of we all had a discussion about this, about, you know, how we should try and keep better, you know, we try and give seminars or something so we know what we're all doing, so we don't waste resources. But of course, then what happened was we started thinking about, well, you know, now we've got two, you know, theirs wasn't quite ready at that point, but it was almost ready to go. Ours was kind of, you know, so but it's not me saying we got there first, so we're taking, you know, it's not an Olympic Games here we're talking about. But what happened was that we then started thinking, well, okay, we've got two of these things now. They're both quite efficient. We both have different interests in using these tools. What can we do with this? And then we started thinking, well, we can just, you know, maybe we can apply them both to, say, human proteins and write a nice little paper on it. And then we started thinking, well, maybe we could do something, you know, and then went a bit further. Eventually, I just said, well, why don't we just do everything? Why don't we just do the whole AlphaFold database and just run these methods? And having both of them will give us a consensus view, so we'll know how reliable these things are, how the predictions are, and we can, you know, the CAF team have expertise in superfamily annotation and things like that. We have an interest in the fold level stuff, perhaps more, and algorithms involving folds, which I'll hope to get time to talk to you a little bit about. And so eventually that was it, and it was kind of one of those things like, you know, well, why not? I mean, how long is it going to take us? It'll only take a month or two to generate all this data. We'll just, you know, write off a quick paper, and then that'll be great. Of course, you know, a year or 15 months later, I mean, when we finally produced the prototype, whenever it was, you always wonder whether if we'd known how long it would have taken to get it right, or at least get it, you know, more, as you know, it still needs it to be a little bit more right in some areas. But, you know, would we have done it? I don't know. But that's how it started. It wasn't a carefully crafted plan where we always, this is something we always wanted to do. But ultimately it worked out very well. And that's how things work sometimes, particularly in academia. Serendipity is a very powerful force in steering science. So the Rengo Labs version with Jude's method here was slightly different from ours. It used a different machine learning approach. It was still trained on CAF, ours was, but it was based on convolutional nets or residual convolutional nets. And so it treated the problem as a kind of an image processing problem, and looking at things like whether residues were in the same secondary structure and how close residues were. And then it used partitioned this 2D matrix to produce predictions. And when we look at the two methods now, and we've looked at it on large amounts of data, they're both very effective methods, but they both you know, have different biases and make mistakes in different places. And so it was quite clear that a consensus approach was going to be better. a consensus between two methods isn't always very good. So we picked another method, which wasn't one of ours, called Unidoc from the group, from the Yang group, the reference at the bottom there. Kind of similar in some, perhaps a little bit similar to some aspects of the chainsaw method, but this is based only on residue on secondary structure units, not on residues. So it's very, very fast for that reason, but can't cut things quite as accurately as the other two methods can. But it still gives a very nice third person in the consensus group, in the committee, that we can use. And so also it's extremely fast, so it was very easy for us to make use of this to add extra methods. Perhaps in the future we'll be making a consensus of 5 methods, or who knows? I mean, who knows? But for now, it's just these 3 approaches. So Andy, I guess it's mostly Andy here, but I guess in discussion with the rest of us, came up with an approach to this. Is an area I think it was work. This is kind of stuff that I think we will focus more work, because I think it's a very interesting problem. Is how do you take very disparate predictions from 3 methods and come up with a consensus? And I think since we've published the paper, we've got a list of ideas that we want to try, including using a quality metric, which I'll very briefly mention towards the end, that we've been defining to try to come up with a single quality estimate of the likelihood that this is a real domain. And I think that's another challenge that I think we're up for over the next year or so. So this method, works pretty well. We take the 3 outputs, we compare them, we work out which bits are they agreeing, which bits are they disagreeing. In the worst case scenario, of course, they just completely disagree with each other, and that ends up then with a low confidence prediction, which we don't consider. Medium confidence is essentially where 2 of them agree, and then we have high confidence if all 3 methods are essentially predicting the same domain boundaries for that particular domain. So it's a very simple approach. I'm not going to go into too much detail here. It's a fairly belt and braces approach. That's not a criticism. It's just often those things that you can get them to work very effectively, very quickly. And you can see more details in the paper if you want to. So now we had our domains. Obviously, we then wanted to classify them. This is obviously where having the input of the CATH team is extremely helpful here, and their long experience of doing this kind of thing on PDB and also in the Gene3D data. Of course, what was also new around the time was the fold-seq method from Martin Steinegger, which is we've used extensively in the CATH project. Certainly in the early stages of it, and later on, as I say, we found some limitations. Some of it, by default at least, the fold-seq was making mistakes, and on the large scale we were working on, there were too many mistakes, and the CATH team were very careful, and as they should be, that they don't want to release incorrectly labeled protein domains. And so I think I set about optimizing the settings of the first step. We used essentially a genetic algorithm to search for optimized settings that reduced actually produced lower false positives and higher true positive rates. Actually, I changed that. I can see I've not saved the edited version of that. That's annoying. I actually was optimizing the F1 score for the labels. So we noticed that it was missing some things that were obvious to us, and we could find them with other tools like the SAP tool that the CATH team used. And so we looked for some other tools that would help fill in some of those gaps. So on the one hand, we wanted to reduce the false positive rates when we were trying to assign superfamilies. Certainly that's important. And also we didn't want to miss too much at the other end. And so although fold-seq, we couldn't have done the work without fold-seq, things like very fast clustering and things like that, it did need a few supplemental tools to be developed. And that's something we had to do. This is the overall view of the picture. I mean, I could probably spend an hour going through everything here, but I'm not going to. You've got the basic idea of it. We took the sequences from the AFDB. We then generated we generated the domains, and we actually in the paper split it into 2 sets. Going forwards, it's going to be just one single set. For the analysis that we all the things that we looked at in the paper, we wanted to reduce the redundancy in the data, and so we only looked at a 100% sequence identical non-redundant subset of the data and focused our analysis on that. But we also filled up the rest of the redundant sequences that were left. They were processed and are processed, and although we currently keep them separate, at least in the paper they're kept separate. From going forwards, it'll just be TED, and there won't be any non-redundant subset. What's very interesting, actually, if you look at the apparently non-redundant of the redundant subset that are identical in sequence to some of the things in TED 100, there are, and you can read the paper, the supplementary material there for more details on that, there are quite surprising differences in the AlphaFold predictions for identical sequences, which is a little bit surprising, but of course the different random number seeds we used, and presumably these were borderline predictions, and we've got lots of interesting case studies of kind of the blooper reel of AlphaFold predicting some things at very low confidence predictions, but I'm not going to bore you with all of that. So the redundancy was actually quite interesting. It allowed us to evaluate this kind of thing, and I think going forwards, I think we can talk to the EBI a bit more about how we could actually maybe detect those kind of changes and try to decide whether they are algorithmic errors, mistakes in the pipeline, or whether these are biologically alternate conformations, for example, which would be very interesting indeed. So from there on, it was a mixture of various levels of clustering using fold-seq and various well-crafted kind of thresholds that we optimized using a genetic algorithm to decide on what combination of coverage, e-value, TM score, various other things would reliably reproduce the cath labels on a what called gold standard set that the cath team calibrate tools on. And so we were able to cover quite a lot of ground quite quickly by just very fast fold-seq searches and mapping them onto the cath data. However, that didn't cover everything, and things were left. We further clustered the sequences of the domains and made sequence-based clusters, and eventually ended up with a number of clusters which went into various directions of processing, some of which I'll look at in the next bit of my talk. And ultimately, we'll get to this kind of the sharp end. I'm not sure if it's the top end of a pyramid or the sharp end of a wedge. I don't know the direction you want to put it in. How we start out with 214 million things, that increase that to a total of about 365 million things, and end up with a list of 7,000 things, which I'll tell you what they are and what you can get from those in a moment. So, right, so a few things, I guess, just general bookkeeping structure. The AlphaFold database finds more domains. I guess that shouldn't really surprise us. If we compare the domain counts in the whole of TED, again, 365 million, that's about 100 million or so more than you can find with Gene3D, and more again from PFAN domain matching. And so that's kind of showing the benefit here. It's not just finding the same old sequence domains again. We're finding more domains than that, and so this is an important upside, I think, to using a structure-based approach to mining out more of these domains, as I've probably alluded to earlier on. Other things, certainly many of them, but not all of these domains come from very high quality AlphaFold models, on PLDDT. Most of them, the ones that aren't in that category, fall down to the high confidence category, and actually relatively small number actually. And I guess this is indicating that our domain finding algorithm is doing a good job here, because obviously if our domain finding algorithms that specifically are trying to, one of the things they're trying to do is discard these low confidence spaghetti regions, we shouldn't be finding very many in this category, and that's reassuringly what we see. So the domains are being defined in the high confidence regions. Interestingly, we do find domain-like things in the low confidence regions and the low confidence. It's not, so they look visually like domains, but because they are the underlying model of low confidence, we can attribute it perhaps to hallucinations from the AI model. We might come back to that later on, I don't know, but hallucination is always a worry when you're using a pure AI method. You don't know whether it's actually just imagining something that doesn't exist or not. And so we exclude everything. In fact, most of the analysis we do, we only use the very top bin of quality in doing the analysis that we do. So we're only using the model, the domains that are very well defined according to AlphaFold, and also very well defined according to our domain parsing algorithms. Again, one thing I didn't mention on that orange bar chart was, in fact, only 5% or roughly 5% of the current data of the AlphaFold database has no domains definable at all using TED, the TED approach. There's this shows you divisions between, say, single domain proteins. The majority of AlphaFold database chains are multiple domains. And this also shows you, although the majority of those multiple domain structures are continuous domains, in other words, they have a single linker between each of the domains that are defined, there are nonetheless, and these are the hardest ones for the parsing methods to find, are discontinuous. So a domain will go into another domain, come back out again, come back perhaps another domain, and you have this very complex topology between the domains. Architecture of domains is discontinuous. So you see not just color block, different color block, but you have kind of blocks and then another color and then back to the original color. That kind of painting structure is what you find quite commonly. And the length distribution, again, most of our domains in TED are shorter than 400 residues, but there are some very big ones. There are always outliers, and some of them are getting on for the 600 to 700, 800. And they're similar to things we've seen before, but otherwise there are very large domains. And we're quite pleased to see that the method wasn't limited to small domains only. So we're able to find a good distribution similar to PDB. So domain classification, I already talked about this a little bit, and I guess in fact, this is the version of the slide I wanted to use before, the geo-optimized settings, but again, it's the same thing. Obviously, we wanted to add some extra tools for this, and I'm going to say now a little bit more about that in a second, what extra tool was developed for that. Obviously, when we look at the labeling that came out of this process, there's a lot of, and again, I don't spend too much, there's two questions being looked at here. How many clusters of sequences are there? So, so for this, we look at the distribution of cluster sizes, the number of clusters. We see most clusters are on the smaller end. That's not so surprising. There are a few very large cluster sizes. And obviously, there's this kind of exponential distribution saying that, for the smaller clusters, there are a lot of those. So a lot of kind of singletons and very small clusters. And then this kind of curve dropping down. Nothing here is that surprising. That's kind of what we would expect to see. The distribution of what we call singletons, in other words, sequences or domain sequences that we can't easily assign to a sequence cluster. There are more of those singletons than there are non-singletons in these clusters. So the clusters are obviously counting for very many more of those sequences, don't forget. So it's not like it's the number of sequences that a singleton is more. It's just the number of sequences, it's just the number of those clusters. We assume each cluster at the sequence level, we can kind of classify roughly at the same label for what fold or superfamily it's in. Not always, but we can maybe assume that. And obviously the singletons are kind of an interesting case because they seem to be maybe new families that haven't been yet explored. So moving on, if we look at that at the superfamily level now, we can say, well, I never try to spend too long on plots like this because I do feel that a lot of this is down to the mechanics of what structures are chosen to solve in PDB and things like that. But there are some things I'll just quickly say. Obviously, there are many more membrane proteins in TED than there are in PDB and CAF. That's for very obvious reasons. There are other structures that are overrepresented in PDB, and that's quite clear from some of these. These are the top folds in these two distributions. But again, I'm not going to spend a lot of time. And again, I could on in a less general talk get more into this because a lot of this, it's unclear whether these are biological differences. One might look at TED and say this is a more reasonable biological distribution, perhaps, of the sizes of these superfamilies. But again, even there are biases depending on which organisms, which have been sequenced, and also which can be predicted by AlphaFold, and which of them have domains that we can find. So there are differences certainly in the breakdown. Some of them are very obvious. Some of them are interesting. A lot of them, I think, just come down to variations according to the purpose of generating that structure. This again is not work that I did. Bashali and Nico worked. So we wanted some case studies. This was. I'm not going to go into the details of this. I don't have the time, but this is a nice example where we were able, by using the TED resource, we were able to trace back the histories of some domains back to organisms that are disease-causing organisms, and we were able to make some interesting functional extrapolations from that, suggesting potential. I mean, obviously, we're long way from the kind of work that we do, but potential kind of directions in which perhaps drugs could be targeted to some of these diseases, these pathogenic organisms. And so this is kind of, it was a very nice study. It's in the paper if you want more details on it. But again, it's also a side of the thing that Christine's team are very expert at and do very well and understand this, have all the tools to analyze it. My tool, we're more at the kind of general, algorithmic level. So it was a very nice case of being able to show the value of the TED resource using a very interesting, set of biological case studies that went into the paper. Another thing, I guess, just going back to the reason we started all of this, Sean in my lab here, we've been interested in work we've done with Pfizer and other projects in looking at how domains pack together. And so Sean has done, again, I don't have time to do justice to this, and I'm going to give you a very quick view of it. But the important thing about this is that this study really can't be done with just defining domains using something like Gene3D or PFAM, because what we're looking at here is the conservation, or at least the distribution of three-dimensional packing or docking of domains in the same chain. So we're looking not just at domain architecture, in other words, what domains are present, but how the domains actually come together, and what angles and what kind of packing arrangements are made. This can only happen if you have the three-dimensional structure of the whole chain with the domains modeled in situ. And so a lot of those structures may not be right, and the main decider here is a thing called the PAE score. And so Sean was very careful only to look at, and the PAE scores are kind of like an RMSD score in a sense, to look at things that had low PAE scores, which are low variation. So we could be more confident, and also the other basic check is to make sure they're actually touching each other. I shouldn't forget that bit. So are these two domains actually packing together, and do they have a low PAE score, which gives the kind of accuracy of the distances between those two independent parts of the chain? And if you see a low value there, then those show that they're probably very well modeled by AlphaFold. And so we did some very nice graphics, to kind of try and show this. I think we'd almost made an album of these things showing the packing of multi-domain proteins. We were able to discover about 10,000 new superfamily interactions that weren't in PDB, and so so these were new interactions between, although they were known domains, they really were in superfamilies that were known. We didn't have an example of them interacting together in the same chain. So this was new data that the AlphaFold database provided that PDB and PDB and Cath didn't have access to. And even if we'd used Gene3D, which wouldn't be able to model the interdomain interactions, that wouldn't have helped either. So this is brand new data that Sean was able to mine out, and he had very nice ways of showing like the increased pairings of certain domains in certain superfamilies that he displayed in these very elegant plots here. This is just one example, another again in the paper, but just showing we have the same reference domain here, and we're showing now, this is what was in PDB or Cath, and this is the same reference structure in grayed out here, and we can see in the AlphaFold database reliably other domains, other superfamilies docking in particularly looking quite realistic packing arrangements. Obviously, all these are hypotheses. I guess experimental structure determination would be probably needed to confirm these things, but very nice combinations of brand new interactions, as I say, that haven't been seen before, and yet they seem to very plausibly appear in the AlphaFold database. And a combination of TED and some of the code that Sean wrote was able to extend this. We've since been following that up. We're now able to massively expand this data set, and so that's some work that we're currently doing to make a very much larger data set of these reliably interacting domains, and not just limiting ourselves to superfamilies, but other proteins as well. And so we're very much looking forward to getting to analyzing that data and in various ways. So finally, I'll get to the sharp end of it, I suppose. I'm running out a little bit of time, so I won't waste too much time on this. I wanted to, at the end of it, again, the thing that we were always hoping to do was to work out about novel folds. And I was looking for a quote, a pithy quote that I could say about the difficulty of defining folds in general, and certainly novel folds. And these are some nice quotes that I got. You're welcome to use any of these. You can attribute them to me if you like. But actually, all I did was I asked ChatGPT to give me some quotes, and I just picked the ones that sounded the best. I'm not going to read them all to you, but I think some of these are quite nice to force proteins into rigid boxes. It's a stray from the gospel of nature's continuum. These all sound like someone much smarter than me and much better read than me has written these poetic things. But anyway, this just to illustrate the point that finding novel domains has always been a thing that we've kind of avoided doing, because it's always been the thing that we shouldn't be doing, because we're not even sure how to define domains because of the continuum. And then when you get to the novel domains, if you can't do that, why are you even trying to count the number of domains? And I will just say, to carry on with this is just to say, it didn't stop us doing it. I was still curious to know what would happen. We also assumed, I think rightly, that the reviewers would ask us to do this, or other people would ask us to do this. So we did it. And again, I'm not going to go through the details of the mechanics here. This is, when you hear a talk, it's better for me just to give you my philosophical view of the thing rather than taking you through the same thing you can read in the paper. I'll just highlight a few things. We did a lot of clustering, again, initially with FoldSeq. Later on, we started using tools of our own to define this. We split very early on, again, using a tool called SIMD. The reference is it's not a tool that we developed, it's from BK Lee's lab. Very early on, we realized that these would either be false positives if we didn't remove them, but also it was very clear that when we started finding these things and showing them, and we showed them to people, everyone went, oh, wow, this is fantastic. And of course, that's just our little simple 8 brains reacting to anything that's symmetric. Of course, these things, all look too beautiful to be wrong, and that would be a mistake if we always assume that. Symmetry is something, of course, we're evolved to recognize and like. And of course, these are amazingly impressive structures. I'm hoping the majority of these will be right and will be determined experimentally. But nonetheless, they are still, they do make fantastic pictures. And there's a lot of things here that some of these have also been looked at in other papers, so we're not the first people to show them. But overall, this is a very impressive, it's very fun show. And you can kind of see the variety of things that AlphaFold has generated here. If we go beyond the non-symmetric that looks pretty, what do we end up with? What's the rest of it look like? Using a staged approach, I worked quite a few months on this myself, actually, because this was my interest. We defined a few things. We defined a new tool, in fact, that I need to rush ahead a little bit so I don't run out of time. So I'm going to say what we did essentially was to ultimately define it very mathematically. We just said a novel fold is something that has a TM score less than 0.5 to any domain that's currently in Cath, Scott, or ECOD. And we could do that initially with FoldSeq, but we also used some of our own tools to do that. And ultimately, we tried to define something called the novelty. And again, I'm going to have to rush a little bit, so I cover and tell you a little bit about where that came from. But it's defined as the average embedding distance between the domain in question and the 20 nearest neighbors, if you like, in the library. And the question is, well, where does this embedding distance come from? One of the tools we developed to increase the range of the TED annotations was a tool I developed first about 5 years ago, just as a proof of concept, just as a learning example I was interested in. And we used an equivariant graph network from the Welling lab to initially to recognize Cath classes. So could we recognize the Cath folds, but from just the C alphas that we fed into this graph model? This type of equivariant model doesn't take rotational symmetry. It doesn't differentiate between mirrors. So this is not a fully equivariant model in the sense of the IPA model that we've used. It's basically distance based. But for recognizing folds, that's sufficient, as it's turned out. It works very well. We trained this initially as a classifier. We initially trained it to classify the different Cath folds. And eventually what we've ended up using it for is just to embed. And so what we do is train it as a classifier. I don't think I've got the training set on that. I've taken that slide out. And we train it purely as given these C alpha coordinates, which Cath fold should you assign to this thing? It works very well, but I guess we didn't want to trust it. We didn't use it in that way. What we did was we forgot the output and we used the latent space just before the output stage as an embedding of that structure. And by training the network to predict the Cath classes accurately and to make things better, we did it separately for the class architecture and topology, we ended up, with a very good classifier and a very robust classifier, but what we really wanted were these embeddings. And what these embeddings ultimately allow us to do is to take all of the C alpha coordinates of variable length and convert them into one fixed length vector, which we can then calculate the distance between using either Euclidean distance or, better still, cosine distance between them. And this allowed us then to define what we call this novelty score, which is the distance between the embeddings of two domains, one of which is in cath or SCOP or ECOD, and the other is in TED, and that we've extracted from that that we're evaluating for its novelty. And so it gives us a nice continuous score. And again, I'm not going to go into details here, again, in the paper. It allowed us to position the novel folds that we came up into, in this case, looking at sequence cluster size, but also a kind of novelty. In other words, how different does this look compared to things already in PDB and in cath that have been classified? And so here's them those pictures. As you can see, these don't have nice symmetry, apart from this one here, which is probably my favorite one. This is this is my favorite, certainly of the non-symmetric, beautiful ones. There is a symmetry here. This is actually a double helix, like DNA, but of helices, of protein helices. So these helices essentially form a helix going in one direction, and then they curl back and then go in the opposite direction, and obviously end up with the N and C terminus back where they are. I think that's a really beautiful structure, that one. It's showing that, I teach people that the double helix equals DNA, and that's where you find it. And now I can say it's also found in proteins, and here's one that we found in the AlphaFold database. But as you can see, all of these things, cover a great, some of the labels show you which organisms they're found in. Charlie's work for that. And, I guess here you can see that these are not as beautiful as the symmetric structures, and also perhaps less plausible in some cases. And this is, I think, the worry about novel fold definition. Maybe the things that are very similar to existing structures, those are perhaps actually existing folds, but just variations of those things. That's the continuum playing tricks with us. And maybe for the things that are really, really novel, perhaps these are hallucinations from AlphaFold. And so we rank them in order of their novelty, but we are not, I would not want to bet a year's salary, or maybe even a month's salary, in saying we actually believe in every single one of these 7,427 domain folds are going to be naturally occurring new unique folds. There are many, many footnotes on that would qualify that. So that's it. I've got a little bit more to say, but I just know that's just covering most of it, but just getting to the end of the TED bit of it. This is the resource. This is the website that you can access. All the data is now available on Zenodo and will be updated as we add more things to it. It has a user-friendly web portal, so you can actually, if you don't want to download the whole lot of data, it's not that big, but if you don't want to download it, you can browse it using a portal that Ian here has very nicely engineered. And so the thing, you can look at the different domains, you can spin them around on the screen, you can link to various resources, to the original AlphaFold structure and so forth. There's information that you can look at the parsing information, make your own mind up. Perhaps you can see a better way of splitting these domains. Coming soon will be a quality score. One of the things that's always been worrying us, is making sure that we have user understandable quality scores, because obviously not every domain is equal in terms of how confident we are that it's a real naturally occurring domain that is stable and so forth. This is coming soon. I'm not going to say very much about it. It's a simple ad hoc score that I put together that combines different pieces of information. Some of that is how similar is it to an existing domain that's already known. Obviously things like the compactness, how compact is the domain, the e-value or the TM score that matches the original domain that's found in the domain libraries. And ultimately, it seems to work quite well. So we're still in the process of evaluating this, but this just shows you, Nico produced a nice plot just showing you here that, we're just deciding now where, we can make your own mind up here where you can make the cut onto these are very, very reliable domains, very robustly defined, and which ones are less reliably defined. It doesn't mean they're wrong. Maybe there's something really exciting at the lower scoring end, but these are the ones you can kind of start betting, a month's salary. Here you might say we're going to bet a week's salary, and here you might, bet a pound. Also, maybe more than that these are actually correct. But of course, experimental validation is really the answer to that, to determining that further. But this will be available shortly, and it'll be a nice continuous score, 0 to 1, and we'll tell you where we think the safest cutoff is. But only use this, and the component scores might also guide you. So if you're interested in learning things that are not superfamilies but new folds, then you'll want to focus more on the things like compactness, and so we'll give you the ability to do that. Okay, quickly, just for a few minutes, I'm just going to skim through it so I can get some questions. One of the things that we have done since this is to go back to the searching of the data that we produce. And we produced a tool. This is kind of something that came out of the fold class work. So we used an earlier version of this in TED itself to define the harder similarities that fold-seq couldn't match. And so we've developed a tool since the TED paper, the preprints out available now, looking for a way of using the embedding method that I described earlier to accelerate the search for multi-domain protein chains. And so unlike fold-seq, which generally just works on a local similarity basis between the query chain and the chains in the database, this tool takes into account the multi-domain structure, not just of the database, because we've already done that, the TED has already done that, but also in the query sequence. So it automatically parses the query sequence, and then using the embedding techniques I've described, then allows various types of multi-domain searching to be done. To do this, we have the embedding vectors that I've already told you about, but we're not going to use them now to find folds, to find the novelty, to calculate the novelty. We're going to use them to bootstrap structure comparisons. So we're going to use it to rapidly search a pre-compiled database of these embeddings that I've described earlier to make a database that's searchable in a very short time. We can look at the top K hits and superpose them, or use TM-align to generate an alignment. And so this is the basic idea. It's a simple idea, but often simple ideas are the ones that work. One of the reasons that we can do this so efficiently is not down to our efforts, but the efforts of people like Facebook, or Meta as they're called now, who have a lot of interest in vector searching. So in their case, I don't even know exactly what kind of features they're looking at, probably whether you buy a particular type of soda or something like that, as they would call it. But anyway, whatever parameters they are searching, they encode the information as embedding vectors and have developed some very, very efficient code to search even billions or trillions of these database items in a relatively short time. The parsing I've told you a little bit about already, so I'm going to skip this. Obviously, we want to parse the domains of the query structure, and we use Marizo for that, which is the tool that we developed. We may allow other tools to be used, slotted in, but for now, Marizo is the search tool for that. And then in Marizo search overview here, it's quite simple. We take the multi-domain query, we break it up into the separate domains, similar to the way we do in the TED database. We then classify them, with fold class, produce those embeddings, and then we compare that embedding of each of those domains with the whole database of 365 million TED domains that have been pre-embedded in a solid-state drive database. And then we can use things like TM-align to validate the top N hits, however many we want to go down. So I'm going to skip through this to hurry up because you can read the preprint. And the answer is, it's a little bit better than fold-seq in various ways. But I'll highlight one thing about that. This is showing that higher sensitivity, and this is why we used it in TED. So as good as fold-seq is, even in its exhaustive mode, we can still find quite reliable matches, quite good matches that it doesn't find. And so maybe that will change as fold-seq improves. But for now, this embedding and then superposition and alignment process is more sensitive. Particularly if you're in a hurry, it's a lot more sensitive. If you don't mind waiting forever, even then it still produces more correct matches than fold-seq does. What's kind of interesting here, and I've highlighted in red this work, I was quite surprised when Sean told me this, that fold-seq is an amazingly fast program, but actually it does have, there are bottlenecks in it. And when we benchmarked an equivalent query of a multi-domain protein against the same database, fold-seq took 37 minutes to do that query. And if we switched it to its intensive mode, 2 hours and a lot of memory. The Marizo search approach with these embeddings found more hits essentially in only 1.5 minutes, literally just 90 seconds of search time. So this approach is very, very effective, both in finding more hits, but also finding those hits very, very quickly. Of course, we're doing that because we're only generating, we're very quickly filtering out the things that have no chance of matching in a more sophisticated way than the way that FoldSync does it with the string matching. So it still has to match strings, and we just go straight to a position in this latent space, and then using these very fast algorithms from Facebook, can quickly jump to the structure matches that are the nearest neighbors of the query domain. I'm going to skip from that to say, just to finish off on this. The searches are you can split the parameters and search for different combinations. So you can search for unordered domain matches. So it doesn't matter what domains are there, as long as they can be in any order, but we don't care. Discontiguous matches, ordered matches. There are options in the code, the freely available code that you can do to run this. And again, this is a case study of one query structure here that matches something in the TED database and ultimately in the AFDB. And it shows you something that FoldSync wouldn't have found, because the domains are completely reordered from the domains as they are in the model, in the query sequence, in the query structure. And so this tool is, I think, can be very valuable to people who are interested, as many people are, into multi-domain architectures. And so the conclusions, again, I'm going to finish. Efficient searching tools are needed now. We've got these large databases. Thankfully, the big tech companies have been exploring some of this space. I think these new domain-based tools, and this is not going to be the last word in that area, I think will have a lot of impact. And then, this is the preprint. You can look at it and find out more about it. You can now also run jobs on the Cypred server, and acknowledgements of everything I've said at the end. There's the TED team. As you can see, even after the year and a half or whatever we worked on this, we're still talking to each other. A large project like this, that's kind of a surprise sometimes. We're having a nice celebratory dinner. Vishali wasn't able to join us, sadly. I've highlighted Dan here because he's been very vital to the Marizo search part of the talk and developing the web server that you can access via the Cypred site. And I think thank you for your attention.